Welcome, everybody. Good evening. Thank you for coming, and to all of the staff who organized it from our public affairs team. It's really awesome. This is my first time doing this, so we're going to go ahead and get started talking about decoding DNA. Even though, I'm from Long Island, which is, I don't know how many people that work at the lab are from Long Island, but I'm from Long Island. So I don't assume, even because you're sitting right here on Main Street, that you've even gone half a mile down the road to see Cold Spring Harbor, but if you haven't, that's the photo. As you know, we're right across the street on Main Street at the Learning Center. Bring your kids, your grandkids, because summer camps are opening soon. I'm required to say that. But we do a whole bunch of experiences for people of all ages. So it starts in middle school, where students might come to look and transform and do some genetic engineering and create some glowing DNA. We also have high school programs. When I was in high school, I don't know if anybody else here from Half Hollow Hills, that was my high school. There might be a few, I see the hands up there, Thunderbirds, right? We have that. And then we also do a whole bunch of project-based training. We work across the entire island, entire region, to get students thinking about all sorts of careers and jobs in sciences. As I mentioned already, the summer camp, so that's my second commercial there, I don't know the third. And what I spent a lot of time on is also helping educators across the world take some of the advanced things that we're doing in the lab and then break it down and bring it into the classroom. We do a lot of stuff online, and as I said, we're many different places in New York, from Brooklyn, which is our largest center that just opened through Regeneron, and then also around the world, where we have centers. I've taught in China, I've gone and visited things that we've done in Australia, we've got something going on in Nigeria, so we have really a global impact from the type of work that we do. All right, so the little short talk that I'm giving is on decoding DNA and DNA sequencing. So I obviously need to say something about this, and it's very interesting, right? Because DNA is something, even if you don't know what it is, you've seen that double helix shape, and it's something that's cultural, right? People, if I search for DNA, there's stock logos that you can buy if your company is involved in that. They just sort of slap it on, and it's one of the few things, if somebody's talking about science, right? You have a DNA, or you have an atom, or you have a microscope. So biology, speaking as biologists, we have two of the three symbols that have anything to do with science that people think about it. But what I'd like you to also think about DNA is, anybody recognize anything here? Right? There's some things you might recognize, and there's some things you might not recognize. But every single culture and every single time, we have documents that define us, right? If you're an American, probably the "we the people" of the Constitution resonates with you. If you're in Western society, the Mona Lisa resonates with you, right? If you're in Asian society, or if you are reading the Talmud, all of those things could resonate with you because they're documents. They're just so iconic, right? That they resonate with us. But we might not all agree on which ones are the most resonant. But the thing is that each one of us, we have DNA inside of us. It's the document, it's the data, it's the digital information that every single person in this room shares. So it really can be thought of on the level of all of those different things as something that's a really great unifier. Okay, I'm supposed to say at least one or two things, because you may not know very much about DNA, and you don't need to know much more than I'm going to tell you tonight. It's got a shape of this double helix, this twisted ladder that we would always explain. And the other kind of key thing, probably the last fact I need to give you, is that it's made up of the base pairs, right? A, T, G, and C. 4 letters that define that information. A goes with T, G goes with C, and you've already passed like 85% of the quiz that I could give you after. But what I would like to kind of make clear, it's always hard, I think it's always hard to talk to a different generation, to a different place in time, when you have something that was amazing for you to have, and then you show it to your kids, your grandkids, and you're like, right? And why is that? Because they kind of take it for granted, right? So if you were an astronomer, the first telescope was the Lippershey telescope in 1608, and that's what Galileo actually based his telescope on. And if you jump 400 years into the future, that's when you start to get the first space telescopes, like the Kepler. We know right now James Webb is the one that gets all the rage, but it took 400 years to go from looking through a tube with ground glass to actually sending something into orbit and being able to take high quality resolutions, okay? In biology, we made the equivalent leap of being able to go from not being able to examine DNA at all to being able to look at all of the DNA, that happened in about 40 years instead of 400 years. We went from the 1960s being able, finally, we knew that DNA was important. We established that for a couple decades, and certainly in 1954, when Watson and Crick made that first announcement, that they were working in the UK, but actually Watson flew back to Cold Spring Harbor to give the first official talk on that. And that was in the 1950s. But from the first time that we could actually sequence it and know what the letters are, actually the first sequencing was not DNA, it was RNA. But when we first got those first letters, that was in 1965. And then the first major, what we call high throughput sequencing, the ability to sequence lots of DNA at once, was in 2006. There was something in between, Sanger sequencing, which is really important. I won't talk so much about that, but it's a 40-year leap in that instead of 400 that biologists were able to do that. And it's really interesting because today, if you were to print out the genome, there's a nice illustration of a person where they printed out in really, really small letters. But it's so amazing when I'm talking with my students about the genome, which is all of our DNA. I can just go to a website and any gene that I have a question about, I tell them, pick on, what's your favorite chromosome? Number 7, I click on it, and I can go and see that information. And that's something that is within our lifetime. So I have to calibrate my sentences because I'm usually teaching when I'm talking about this 10th or 11th graders, I'm trying to figure out, okay, when were these ones born? And then I'm like, okay, yes, this is in your lifetime, but maybe sometimes it's within their parents' lifetime. This, before 2000, we couldn't do this. Let me say that for 100% sure, that we couldn't come to a website and do that. I want to remind people also that the history of this starts down the street. This is up in Lloyd Harbor at the Banbury Center, where you could see Watson, you could see Crick, you could see, no, not Crick, you could see, what's his name, Collins, because I saw him a couple weeks ago. You could see that this started, the very first meetings planning the human genome started just down the road from you. And then it was a long timeline of about 1,100 scientists to over dozens of countries to do DNA sequencing, because we knew that, hey, if we want to solve things like cancer, if we want to solve genetic diseases, we need to know the whole human genome. And that whole timeline stretches across the late 90s to the early 2000s. 2003 was when they said they finished it. They really didn't. Remember? Well, in 2 slides, you'll believe me that they didn't finish it, but 1 second, let me get to 1 slide before that. What I wanted to point out here, this is a photograph from Baylor College of Medicine, is that in order to sequence the genome, imagine hundreds of sites across the world filled with rooms, filled with these big machines that were doing sequencing. That's what it looked like. It was a massive effort that was $1 billion. So does anybody know how much DNA there is in a human? How many letters? How many A's, C's, T's, and G's? Let me hear a few guesses. Give me a guess. Give me a guess.
5 million.
5 million. This sounds like The Price is Right now. 5 million. Anybody else? Not the scientists who already know the answer. Who else? Who in the back row? You with the folded arms. Who doesn't want to answer? 5 million. Higher, lower?
Much higher.
How much higher? Give me a number.
4 billion.
4 billion. Okay. It's about 3 billion. Okay. 3.2. So you're close, but you were over. You would win, right? The 500. How big, the other thing I always like to ask the question, just when I'm asking with students, how big is a billion, by the way? Okay, well that's more, how old are you when you're first 1 million seconds old?
Oh, God.
When you come back from the hospital, million seconds old, when do you reach your first million
30
seconds?
years.
Huh?
30 years.
No, that's your first million seconds. No, that's a little bit late. First million seconds.
4 years old.
I'm hearing years and hearing things. You're about 11 days old. Okay. Now the question is, when do you reach your first 1 billion seconds?
85.
85 what? I'm a scientist, I want units. Other guesses?
40 years.
A little bit lower, like it's when you're almost 32 years old, right? So in your 30s, right, when you reach your first billion. Do we have anyone who's 3 billion seconds old yet? Maybe not.
Okay, so yeah, so you could say that, right? I'm not even 3 billion, 30 billion something. But it took essentially the cost of sequencing this was $1 a nucleotide, $1 an A, T, C, or G. So it was $3 billion. That was really expensive, right? I told you that they announced that it was finished, but it really wasn't finished. This was the announcement, and so it's it was the official like it's finished, but there were always these little dark pieces of the genome that they hadn't quite finished. But for government purposes, it was finished, right? They had said we spent the money. But the key thing that really matters to everybody in this room is that the cost of doing that has decreased dramatically. So back in, let's say 2000, which is when this chart goes to, it would be $100 million to sequence a genome. So that means if we have a sick kid in the hospital, we don't know what's wrong, we think it might be genetic because we can't figure out anything else, it would be $100 million test. I'm always talking this to the kid, and it's like, do you think your parents would pay the $100 million? Some of the kids actually honestly answer no. But it's $100 million, right? Now, the goal for the last couple of years had been $1,000, and now it's about $600, okay? And with insurance, they probably triple charge you, whatever it could. But that's within the realm of, yeah, okay, I'll be able to do that, right? And it's really clear that, the technology started in the late almost 2010. It's really 2008 that inflection really starts. But imagine that, right? Going from $100 million to $600, right? That means that when we're asking questions about ourselves and about our health and about looking what's going on in the genome, even with cancer, where it's particularly like the thing now is if somebody's got a cancer, we're going to sequence it because we want to know exactly what has gone wrong in that particular cancer. Everyone is different. And because it's become so cheap, we can do that for every single patient, right? That's where we're going with this. One of the technologies that has allowed this is not the only technology, it's just the one I'm going to talk about tonight, is by this company called Oxford Nanopore. As you can tell by the name, it's in Oxford, UK. Don't worry, there were Americans involved in the actual original idea. But the super cool thing about that is that it is not only a super cheap way to sequence a lot of DNA at once, but as you can see, and as I'm going to demonstrate in a moment, it's something I can hold in the palm of my hand, right? You've gone from rooms and rooms and rooms of DNA sequencers and millions or hundreds of millions of dollars to do DNA sequencing to something that I can do with my 12th graders, with my 11th graders, with a bright 9th grader in the morning, the right time of day, right? You got to do that. And that's what we have, right? So it's the power of being able, just like in computing, right? The power of miniaturization allows us to do that. I'm not going to go too deeply into what the technology works, but that opening graphic, credit to the designers at Nanopore, the name of the sequencing actually suggests what it is. It literally is a nano, very small hole or pore inside of a membrane, inside of a device, which I'm going to show you in a little bit live. But what happens is that using the techniques of molecular biology, we can put DNA onto this surface. A little enzyme guides the DNA to the pore, and the pore sucks it down. And as it's going through this little hole, it's creating minute disturbances in the electric charge that's being measured. So for those of you, maybe there's electrical engineers in the audience, it's basically a voltmeter going up and down. But it's measuring the electric charge, and as it turns out, biochemically, that an A is different than a T, different than a G, different than a C, and so they all have their characteristic disturbances to that charge. And that little it's not an EKG, but we call it a squiggle, technical term, on the right is the electrical signal, which essentially gets translated into DNA sequence. Okay, I'm almost wrapping up here. This is amazing for us because at the Learning Center, particularly at Cold Spring Harbor, we've always thought that there's no difference between our students and our researchers. We want them to be using the same tools and the same technology. And we've been doing that for years, right? This is an example of types of setups that we've sent to our kids, whether they're working in their own high schools or during the pandemic when everything was virtual. The ability to also give a creative young student these tools very early allows them to be creative. The picture I always go back to is one that we did. This group in the Bronx, they were one of our first winners of a little project they did on DNA barcoding, where they had gone to CVS, Walgreens, medicine shops, traditional medicine, looking for ginkgo supplements, and they were like, "Hey, let's go see all of these things. It's just a little green pill. Does it really have ginkgo?" Spoiler: no ginkgo in any of the things that they collected, right? And it was confirmed later on by the Attorney General, saying cease and desist to companies, and it's not necessarily the company even knew, right? But if you're not looking at the DNA, right, these things are obscured, and you may not know because all you'll have is a little green tablet. It was rice clippings, like rice. You could imagine probably there's a rice patty, and like, what are you going to do all this rice? We're going to cut it with ginkgo. We've done things that are more sophisticated through that. We work with it the last couple of years, taking our 11th and our 10th graders that come to the summer camps and getting them to sequence genomes, right? So we sequence things the first year in 21, we were looking at duckweed, which others at the lab are developing into biofuels. Last year we did it with an endangered plant that's endemic to Puerto Rico. This last summer, well, that was 2022, 23 was chloroplast DNA. So really, our ability to understand DNA is critical to any kind of problem that we're going to have in biology. It may not be the cause of the problem, but it's in there somewhere in terms of its relevance. And so that's what we do. We look at this thing. So I do have a thank you slide, but before I get to the thank you slide, I'm going to call Chris up here because we're going to try to sequence some DNA for you live. We're going to see how it works. So let me get this set up just a little bit. So let me explain what's happening and show you yourselves, right? So I want to do a close-up on this. If you want to, you can. Everybody wants to see themselves. That's like the most amazing. We do get a chance to see yourself because usually we're looking at people's DNA, but today we are sequencing, of all things, yeast DNA, brewer's yeast, because in a bar, what would you sequence except for some yeast DNA? So I'm going to demonstrate just a little bit of this, and what I should do is take the mic with me. And I'm going to walk over to my DNA sequencing lab while Chris eventually has a use for the camera. What we can take a look at right now is this is one of our DNA sequencing devices. So this is the fancy one. So this is sort of the computer screen, and this is the actual little thing that we call a cell, and maybe we'll pass one of these around, or you can come up later. They're $900. Please give it back to me. But what I'm going to try to do is I'm going to go ahead and try to load some DNA onto it. Oh, let me hold it this way. Oh, that's perfect. So let me try to describe what I'm doing. Okay, so I have my DNA sequencing lab, and I have a whole bunch of things here. Hopefully I brought all the right things. And I need to just mix a couple things in the right amount, which I've written on the back of my hand because I realized I took a photo of what I'm supposed to do, but then I realized if I'm using my camera, I won't be able to use my photo. Let's see if we can get a little bit closer. So it doesn't look like very much. In fact, most of biology is moving clear drops of liquid from one tube in another for hours on end. Okay. Then we've got a little bit of buffer, which is going to control the pH. And then we've got these little microscopic glass beads, and they actually help the DNA to roll across the surface of the sequencing surface. But let me do the mixing first. Let's see, the order I'm supposed to do this one first. Okay. So I've got to mix this, kind of like I'm not as good as the bartender back there, but I have to mix the beads a bit. That's good enough. Okay, so now let's try to load it because this one is worth looking close up a little bit if we can get it to work. All right, so I'm going to go ahead and just open this port here. And now let's see. I'm going to try to do the dropping. I'll put the microphone down for this, and maybe you can zoom in as much as you can. All right. It's going to soak in there. So you can take the camera off for a second. Oh, there we go. A little bit of light helps. And then, that's good enough. Now we basically wait. What it's going to show you for a little while
and we'll come back to it a little bit later, is that it's starting to sequence. So we're going to give that time. And thank you for the camera. We'll come back to it. So yeah, essentially what you're looking at with my shaky camera hand is as the DNAs are passing through the pores, it lights up in this disco thing, so that was very creative of the company to come up with that display. As the sequence is moving through the pores, you're getting all these indicators, and eventually I'll have a bunch of DNA from this particular sample. Yeah, so that's what it kind of looks like. And sorry, there's no actual A's, T's, or G's, but you get the idea. Later on there will be. The last thought there, as I say thanks, that the illiterate of the 21st century will not be those who cannot read and write, but those who cannot learn, unlearn, and relearn. So thank you for coming tonight to learn with us. In the meantime, we'll just take some questions. So thank you.